Hello, Namaste. Myself Ankita Shreya Prasad, and you are listening to along with our NCERT 11th and 12th audio video book presented by ArtSkill Passion. Chapter 12, Mineral Nutrition. The basic needs of all living organisms are essentially the same. They require macromolecules such as carbohydrates, proteins, and fats, and water and minerals for their growth and development. This chapter focuses mainly on inorganic plant nutrition, wherein you will study the methods to identify elements essential to growth and development of plants and the criteria for establishing the essentiality. You will also study the role of the essential elements, their major deficiency symptoms, and the mechanism of absorption of these essential elements. The chapter also introduces you briefly to the significance and the mechanisms of biological nitrogen fixation. 12.1 Methods to study the mineral requirements of plants. In 1860, Julius von Sachs, a prominent German botanist, demonstrated for the first time that plant could be grown to maturity in a defined nutrient solution in complete absence of soil. This technique of growing plant in a nutrient solution is known as hydroponics. Since then, a number of improvised methods have been employed to try and determine the mineral nutrients essential for plants. The essence of all these methods involve the culture of plants in a soil-free, defined mineral solution. These methods require purified water and mineral nutrient salts. Can you explain why is this so essential? After a series of experiments in which the roots of the plants were immersed in nutrient solutions and wherein an element was added, substituted, removed, or given in varied concentration, a mineral solution suitable for the plant growth was obtained. By this method, essential elements were identified and their deficiency symptoms discovered. Hydroponics has been successfully employed as a technique for the chemical production of vegetables such as tomato, seedless cucumber, and lettuce. It must be emphasized that the nutrient solution must be adequately aerated to obtain the optimum growth. What would happen if solutions were poorly aerated? Diagrammatic views of the hydroponic techniques is given in figure 12.1 and 12.2. 12.2 Essential mineral elements. Most of the minerals present in soil can enter plants through roots. In fact, more than 60 elements of the 105 discovered so far are found in different plants. Some plant species accumulate selenium, some other gold, while some plants growing near nuclear sites take up radioactive strontium. There are techniques that are able to detect the minerals even at a very low concentration, 10 to the power minus 8 gram by milliliters. The question is whether all the diverse mineral elements present in a plant, for example gold and selenium as mentioned above, are really necessary for plants. How do we decide what is essential for plants and what is not? 12.2.1 Criteria for essentiality. The criteria for essentiality of an element are given below. A. The element must be absolutely necessary for supporting normal growth and reproduction. In the absence of the element, the plants do not complete their life cycle or set the seeds. B. The requirement of the element must be specific and not replaceable by another element. In other words, deficiency of any one element cannot be met by supplying some other element. C. The element must be directly involved in the metabolism of the plant. Based upon the above criteria, only a few elements have been found to be absolutely essential for plant growth and metabolism. These elements are further divided into two broad categories based on their quantitative requirements. First, macronutrients and second, micronutrients. Macronutrients are generally present in plant tissue in large amounts, in excess of 10 mmol/kg of dry matter. The macronutrients include carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium, and magnesium. Of these, carbon, hydrogen, and oxygen are mainly obtained from CO2 and H2, while the others are absorbed from the soil as mineral nutrition. Micronutrients or trace elements are needed in very small amounts, less than 10 mmol/kg of dry matter. These include iron, manganese, copper, molybdenum, zinc, boron, chlorine, and nickel. In addition to these 17 essential elements named above, there are some beneficial elements such as sodium, silicon, cobalt, and selenium. They are required by higher plants. Essential elements can also be grouped in four broad categories on the basis of their diverse functions. These categories are: first, essential elements as components of biomolecules and hence structural elements of cells. Example: carbon, hydrogen, oxygen, and nitrogen. Second, essential elements that are components of energy-related chemical compounds in plants. Example: magnesium in chlorophyll and phosphorus in ATP. Third, essential elements that activate or inhibit enzymes. For example, Mg+2 is an activator for both ribose bisphosphate carboxylase oxygenase and phosphophenol pyruvate carboxylase, both of which are critical enzymes in photosynthetic carbon fixation. Zn+2 is an activator of alcohol dehydrogenase and MO of nitrogenase during nitrogen metabolism. Can you name a few more elements that fall in this category? For this, you will need to recollect some of the biochemical pathways you have studied earlier. Some essential elements can alter the osmotic potential of a cell. Potassium plays an important role in the opening and closing of stomata. You may recall the role of minerals as solutes in determining the water potential of a cell. 12.2.2 Role of macro and micronutrients. Essential elements perform several functions. They participate in various metabolic processes in the plant cell, such as permeability of cell membrane, maintenance of osmotic concentration of cell sap, electron transport system, buffering action, enzymatic activity, and act as major constituents of macromolecules and coenzymes. Various forms and functions of essential nutrient elements are given below. Nitrogen. This is essential nutrient element required by plants in the greatest amount. It is absorbed mainly as NO3 minus, though some are also taken up as NO2 minus or NH4 plus. Nitrogen is required by all parts of a plant, particularly the meristematic tissues and the metabolic active cells. Nitrogen is one of the major constituents of proteins, nucleic acids, vitamins, and hormones. Phosphorus. Phosphorus is absorbed by the plants from soil in the form of phosphate ions, either as H2PO4- or HPO4-2. Phosphorus is a constituent of cell membranes certain proteins, all nucleic acids and nucleotides, and is required for all phosphorylation reactions. Potassium, it is absorbed as potassium ion K+. In plants, this is required in more abundant quantities in the meristematic tissues, burrs, leaves, and root tips. Potassium helps to maintain an anion-cation balance in cells and is involved in protein synthesis, opening and closing of stomata, activation of enzymes, and in the maintenance of the turgidity of cells. Calcium. Plant absorb calcium from the soil in the form of calcium ions Ca+2. Calcium is required by meristematic and differentiating tissues. During cell division, it is used in the synthesis of cell wall, particularly as calcium pectate in the middle lamellae. It is also needed during the formation of mitotic spindle. It accumulates in ordered leaves. It is involved in the normal functioning of the cell membranes. It activates certain enzymes and plays an important role in regulating metabolic activities. Magnesium. It is absorbed by plants in the form of divalent Mg+2. It activates the enzymes of respiration, photosynthesis, and are involved in the synthesis of DNA and RNA. Magnesium is constituent of the ring structure of chlorophyll and helps to maintain the ribosome structure. Sulfur. Plants obtain sulfur in the form of sulfate, SO4-2. Sulfur is present in two amino acids, cysteine and methionine, and is the main constituent of several coenzymes. Vitamins. Thiamine, biotin, coenzyme A, and ferredoxin. Iron. Plants obtain iron in the form of ferric ions Fe+3. It is required in larger amounts in comparison to other micronutrients. It is an important constituent of proteins involved in the transfer of electrons like ferredoxin and cytochromes. It is reversibly oxidized from Fe+2 to Fe+3 during electron transfer. It activates catalyze enzymes and is essential for the formation of chlorophyll. Manganese. It is absorbed in the form of manganese ions Mn+2. It activates many enzymes involved in photosynthesis, respiration, and nitrogen metabolism. The best defined function of manganese is in the splitting of water to liberate oxygen during photosynthesis. Zinc. Plants obtain zinc as Zn+2 ions. It activates various enzymes, especially carboxylase. It is also needed in the synthesis of oxygen. Copper. It is absorbed as cupric ions Cu+2. It is essential for the overall metabolism in plants. Like iron, it is associated with certain enzymes involved in redox reactions and is reversibly oxidized from Cu+ to Cu+2. Boron. It is absorbed as BO3 3- or B4O7-2. Boron is required for uptake and utilization of Ca+2. Membrane functioning, pollen germination, cell elongation, cell differentiation, and carbohydrate translocation. Molybdenum. Plants obtain it in the form of molybdate ions MoO2+2. It is a component of several enzymes including nitrogenase and nitrate reductase, both of which participate in nitrogen metabolism. Chlorine. It is absorbed in the form of chlorine ions Cl- along with Ca+ and K+. It helps in determining the solute concentration and the anion-cation balance in cell. It is essential for the water splitting reaction in photosynthesis, a reaction that leads to oxygen evolution. 12.2.3 Deficiency symptoms of essential elements. Whenever the supply of an essential element becomes limited, plant growth is retarded. The concentration of the essential element below which plant growth is retarded is termed as critical concentration. The element is said to be deficient when present below the critical concentration, since each element has one or more specific structural or functional role in plants. In the absence of any particular element, plants show certain morphological changes. These morphological changes are indicative of certain element deficiency and are called deficiency symptoms. The deficiency symptoms vary from element to element and they disappear when the deficient mineral nutrients is provided to the plant. However, if deprivation continues, it may eventually lead to the death of the plant. The parts of the plant that show the deficiency symptoms also depend on the mobility of element in the plant. For example, elements that are actively mobilized within the plants and exported to young developing tissues. The deficiency symptoms tend to appear first in the older tissues. For example, the deficiency symptoms of nitrogen, potassium and magnesium are visible first in the senescent leaves. In the older leaves, biomolecules containing these elements are broken down, making these elements available for mobilizing to younger leaves. The deficiency symptoms tend to appear first in the young tissues whenever the elements are relatively immobile and are not transported out of the mature organs. For example, elements like sulfur and calcium are a part of the structural component of the cell and hence are not easily released. This aspect of mineral nutrition of plants is of a great significance and importance to agriculture and horticulture. The kind of deficiency symptoms shown in the plant include chlorosis, necrosis, stunt plant growth, premature fall of leaves and burrs and inhibition of cell division. Chlorosis is the loss of chlorophyll leading to yellowing in leaves. This symptoms is caused by the deficiency of elements nitrogen, potassium, magnesium, sulfur, iron, manganese, zinc and MO. Likewise, necrosis or death of tissues, particularly leaf tissues, is due to the deficiency of calcium, magnesium, copper, potassium. Lack or low level of nitrogen, potassium, sulfur and MO causes an inhibition of cell division. Some elements like NSMO delay flowering if their concentration in plants is low. You can see from the above that the deficiency of an element can cause multiple symptoms and thus the same symptoms may be caused by the deficiency of one of several different elements. Hence, to identify the deficient element, one has to study all the symptoms developed in all the various parts of the plant and compare them with the available standard tables. We must also be aware that different plants also respond differently to the deficiency of the same element. 12.2.4 Toxicity of micronutrients. The requirement of micronutrients is always in low amounts, while their moderate decrease causes the deficiency symptoms and a moderate increase causes toxicity. In other words, there is a narrow range of concentration at which the elements are optimum. Any mineral ion concentration in tissues that reduces the dry weight of tissues by about 10% is considered toxic. Such critical concentration vary widely among different micronutrients. The toxicity symptoms are difficult to identify. Toxicity levels for any element also vary for different plants. Many times, excess of an element may inhibit the uptake of another element. For example, the prominent symptom of manganese toxicity is the appearance of brown spots surrounded by chlorotic veins. It is important to note that the manganese competes with iron and magnesium for uptake and with the magnesium for binding with enzymes. Manganese also inhibits calcium translocation in shoot apex. Therefore, excess of manganese may in fact induce deficiencies of iron, magnesium and calcium. Thus, what appears as symptoms of manganese toxicity may actually be the deficiency symptoms of iron, magnesium and calcium. Can this knowledge be of some importance to a farmer, a gardener or even for you in your kitchen garden? 12.3 Mechanism of absorption of elements. Much of the studies on mechanism of absorption of elements by plants has been carried out in isolated cells, tissues or organs. These studies reveal that the processes of absorption can be demarcated into two main phases. In the first phase, an initial rapid uptake of ions into the free space or outer space of cell. The apoplast is passive. In the second phase of uptake, the ions are taken in slowly into the inner phase. The symplast of the cells, the passive movement of ions into the apoplast usually occurs through ion channels. The transmembrane proteins that functions as selective pores. On the other hand, the entry or exit of ions to and from the symplast requires the expenditure of metabolic energy, which is an active process. The movement of ions is usually called flux. The inward movement into the cell is influx and the outward movement efflux. You have read the aspects of mineral nutrients uptake and translocation in plants in chapter 11. 12.4 Translocation of solutes. Mineral salts are translocated through xylem along with the ascending stream of water, which is pulled up through the plant by transpirational pull. Analysis of xylem sap shows the presence of mineral salts in it. Use of radioscopes of mineral elements also substantiate the view that they are transported through the xylem. You have already discussed the movement of water in xylem in chapter 11. 12.5 Soil as reservoir of essential elements. Majority of the nutrients that are essential for the growth and development of plants become available to the roots due to weathering and breakdown of rocks. These processes enrich the soil with dissolved ions and inorganic salts. Since they are derived from the rock minerals, their role in plant nutrition is referred to as mineral nutrition. Soil consists of a wide variety of substances. Soil not only supplies minerals but also harbors nitrogen fixing bacteria. Other microbes hold water supplies aid to the root and act as a matrix that stabilizes the plant. Since deficiency of essential minerals affect the crop yield, there is often a need for supplying them through fertilizers. Both macronutrients, nitrogen, phosphorus, potassium, sulfur, etc. and micronutrients, copper, zinc, iron, manganese, etc. form components of fertilizers and are applied as per need. 12.6 Metabolism of nitrogen. 12.6.1 Nitrogen cycle. Apart from carbon, hydrogen and oxygen, nitrogen is the most prevalent element in living organism. Nitrogen is a constituent of amino acids, proteins, hormones, chlorophylls and many of the vitamins. Plants compete with microbes for the limited nitrogen that is available in soil. Thus, nitrogen is a limiting nutrient for both the natural and agricultural ecosystems. Nitrogen exists as two nitrogen atoms joined by a very strong triple covalent bond. NN triple bond. The process of conversion of nitrogen to the ammonia is termed as nitrogen fixation. In nature, lightning and ultraviolet radiation provide enough energy to convert nitrogen to nitrogen oxides. NO, NO2, N2O. Industrial combustion, forest fires, automobile exhaust, and power generating stations are also source of atmospheric nitrogen oxides. Decomposition of organic nitrogen of dead plants and animals into ammonia is called ammonification. Some of the ammonia volatiles and re-enters the atmosphere, but most of it is converted into nitrate by soil bacteria in the following steps. NH3 plus O2 give rise to NO2 plus H plus H2. NO2 plus O2 give rise to NO3. Ammonia is first oxidized to nitrate by bacteria, nitrosomans and or nitrococcus. The nitrate is further oxidized to nitrate with the help of the bacterium Nitrobacter. These steps are called nitrification. Figure 12.3. These nitrifying bacteria are chemoautotrophs. The nitrate thus formed is absorbed by plants and is transported to the leaves. In leaves, it is reduced to form ammonia that finally forms the amine group of amino acids. Nitrate present in the soil is also reduced to nitrogen by the process of denitrification. Denitrification is carried by bacteria, Pseudomonas and Theobacillus. 12.6.2 Biological nitrogen fixation. Very few living organisms can utilize the nitrogen in the form of N2 available abundantly in the air. Only certain prokaryotic species are capable of fixing nitrogen. Reduction of nitrogen to ammonia by living organisms is called biological nitrogen fixation. The enzyme nitrogenase, which is capable of nitrogen reduction, is present exclusively in prokaryotes. Such microbes are called N2 fixers. NN triple bond give rise to NH3 in the presence of nitrogenase. The nitrogen fixing microbials could be free living or symbiotic. Example of free living nitrogen fixing aerobic microbes are Acetobacter and Bencheringia, while Rhodospirillum is anaerobic and free living. In addition, a number of canobacteria such as Anabania and Nostoc are also free living nitrogen fixers. Symbiotic biological nitrogen fixation. Several types of symbiotic biological nitrogen fixing associations are known. The most prominent among them is legume bacteria relationship. Species of rod species Rhizobium has such relationship with the roots of several legumes such as alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean, clover beans, etc. The most common association on roots is as nodules. These nodules are small outgrowths on the roots. The microbe Frankia also produces nitrogen fixing nodules on the roots of non-leguminous plant. Example, Alnus. Both Rhizobium and Frankia are free living in soil, but symbionts can fix atmospheric nitrogen. Uproot any one plant of a common plus just before flowering. You will see near spherical outgrowth on the root. These are nodules. If you cut through them, you will notice that the central portion is red or pink. What makes the nodule pink? This is due to the presence of leguminous hemoglobin or leg hemoglobin. Nodule formation. Nodule formation involves a sequence of multiple interactions between rhizobium and roots of the host plant. Principal stages in the nodule formation are summarized as follows. Rhizobia multiply and colonize the surroundings of roots and get attached to epidermal and other root hair cells. The root hair curl and the bacteria invade the root hair. An infection thread is produced carrying the bacteria into the cortex of the root, where they initiate the nodule formation in the cortex of the root. Then the bacteria are released from the thread into the cells. which leads to the differentiation of specialized nitrogen fixing cells. The nodule thus formed establishes a direct vascular connection with the host for exchange of nutrients. These events are depicted in figure 12.4. The nodule contains all the necessary biochemical components such as enzymes, nitrogenase and like hemoglobin. The enzyme nitrogenase is a MOFE protein and catalyzes the conversion of atmospheric nitrogen to ammonia. Figure 12.5. The first stable product of nitrogen fixation. The reaction is as follows: N2 plus 8 electrons plus 8H plus 16 ATP raised to 2 NH3 plus H2 plus 16 ADP plus 16 Pi. The enzyme nitrogenase is highly sensitive to the molecular oxygen. It requires anaerobic conditions. The nodules have adaptations that ensure that the enzyme is protected from oxygen. To protect these enzymes, the nodule contains an oxygen scavenger called like hemoglobin. It is interesting to note that these microbes live as aerobes under free living condition, where nitrogenase is not operational, but during nitrogen fixing event they become anaerobic, thus protecting the nitrogenase enzyme. You must have noticed in the above reaction that the ammonia synthesizes by nitrogenase requires a very high input of energy, 8 ATP for each NH3 produced. The energy required thus is obtained from the respiration of the host cells. Fate of ammonia. At physiological pH, the ammonia is protonated to form NH4 plus ammonium ion. While most of the plants can assimilate nitrate as well as ammonium ions, the latter is quite toxic to plant and hence cannot accumulate in them. Let us now see how the NH4 plus is used to synthesize amino acids in plants. There are two main ways in which this can take place. First, reductive aminization. In this process, ammonia reacts with alpha ketoglutaric acid and form glutamic acid as indicated in the equation given below. Alpha ketoglutaric acid plus NH4 plus NADPH give rise to glutamate plus H2O plus NADP in the presence of glutamate dehydrogenase. Second, transamination. It involves the transfer of amino group from one amino acids to the keto group of keto acid. Glutamic acid is the main amino acid from which the transfer of NH to the amino group takes place, and other amino acids are formed through transamination. The enzyme transaminase catalyzes all such reactions. For example, the two most important amides, asparagine and glutamine, found in plants are structural part of proteins. They are formed from two amino acids, namely aspartic acid and glutamic acid, respectively by addition of another amino group to each. The hydroxyl part of the acid is replaced by another NH2 minus radical. Since amides contain more nitrogen than the amino acids, they are transported to other parts of the plant via xylem vessels. In addition, along with the transpiration stream, the nodules of some plants, example soybean, export the fixed nitrogen as urides. These compounds also have a particularly high nitrogen to carbon ratio. Thank you for being till the end. I hope this has helped you. If so, give thumbs up, like, share, comment, save, download and subscribe. You can support us by following in other platforms. That link is in the description. Well, thank you. See you later. Maybe never.